segmentation of membranes by heterogeneous environments. In cells, the environment on one side of a membrane is not uniform. It changes in space, and those changes alter how membrane lipids and proteins interact with their surroundings. Here is a cartoon of a plasma membrane with its actin cortex below. The membrane contains many species of lipids and embedded proteins. Different molecules have different affinity for cytoskeletal proteins. As a result, the membrane is effectively divided into segments, where certain lipids or receptors are enriched while others are depleted. How do we detect such segmentation? Single particle tracking follows one labeled molecule over time. This trajectory shows confined diffusion inside one segment, then an occasional hop into a neighboring segment, and again confined motion. In one classic experiment with a 40 nanometer gold tag on a transferrin receptor, the average segment radius was about 280 nanometers, and the average residence time in a segment was about 29 seconds. What causes confinement? Two classic ideas are fences and pickets created by the actin cortex. Fences are essentially immobile cytoskeletal fibers that coral proteins with cytosolic domains. Pickets are transmembrane proteins that transiently bind the cortex, forming rows that impede even lipids in the outer leaflet. Sometimes, receptors are attracted to actin tracks and diffuse almost one-dimensionally along the mesh. The cell cortex is complicated, but the same idea appears in simpler systems, supported membranes or pore-spanning membranes that experience two different environments. The two sides impose different molecular fields on adjacent segments. In a single component membrane, only a tiny change in density occurs. In a multi-component membrane, composition also changes. A key theoretical result is that phase domains can form only in one segment at a time, not in several segments simultaneously. Inside living cells, the environment varies from segment to segment, and over time, different contact segments with distinct actin-binding proteins, and non-contact segments with transient scaffolds like clathrin. The membrane is partitioned into many segments, each with its own local field. If those local fields act like long-lived random fields in 2 dimensions, they can prevent large domains from forming, similar to the 2-dimensional Ising model with random fields. Instead of one big domain, you see only small fluctuating patches. Summary: heterogeneous environments segment membranes into regions with different local interactions. Single particle tracking reveals confined diffusion with occasional hops between segments. Cytoskeletal fences and pickets impede lateral motion, and some receptors slide along actin. In multi-component membranes, phase domains appear in one segment at a time. In highly heterogeneous cellular environments, long-lived random fields can suppress macroscopic phase separation.